Aggressive behavior is a very common in animal kingdom. It exists in apes, other mammals, and invertebrates, or even fruit flies. It is used to defend resources such as food or mate. And despite that it is a common behavior, the genetic basis of aggression is still poorly understood. In our lab, we developed fly model of aggression and tried to gain insight into its basic mechanism. In our lab, we used two different fly lines. The top video shows a typical fight between two wild-type Cantonese males. They engage in the regular lunge behavior. The bottom video shows a fight between two bully flies. While they are fighting, I will show you some quantification of the behavior. As you can see, bully displays more lunge behavior and have a much higher percentage of fight showing boxings. And this is what they are engaging in the boxing behavior. To identify the genetic basis of this behavioral difference, we use RNA sequencing to look at the transcriptome in these flies. We compare 4 different samples: wild-type males that raise at 19 degrees Celsius or 25 degrees Celsius versus bully, the hyper-aggressive line that raise at 25 degrees Celsius and 19 degrees Celsius. We extract RNA from the fly head and perform sequencing using Illumina Hi-C. From the sequencing result, we have about 30 million reads for each sample. Then we map and count the sequence read using software like TopHat and genome features. And finally, to compare the result and identify the differentially expressed genes, we perform statistical analysis using HR. The resulted data will be analyzed using Spark and Neo4j. In this project, I will demonstrate the use of Sparks and Neo4j to analyze RNA-seq data and identify candidate genes that affect aggressions. First, I load the differential genes data into the Spark data frame. There are over 13,000 genes per each experimental conditions and 4 conditions in total. Here is one example of gene data when we compare bully raised at 19 degrees Celsius to wild-type Cantonese raised at 19 degrees Celsius. There are a total of 7 columns. The first one is the gene ID number. Base mean is the average of normalized count for all samples. Log two-fold changes is the log fold change. If it is +1, that means it is two-fold more in bully. Log fold change standard error is just the standard error. Stat and the p-values is the raw statistic we use to calculate significance. However, since we have over 13,000 genes, p-value of 0.05 will give us too many false positives. To account for that, we use the Benjamini-Hochberg adjust p-value for comparison. For simplicity, I load all 4 datasets in a list, and here is the schema. As you can see, most of the columns are in fact string. which I need to convert them to double. Also, as you remember in the previous slide, some rows contain Na. Since not all genes are expressed in the cells, genes with low WickBunChan can have Na in some columns. I also need to remove them. In Spark, I can easily change the column type and drop the rows with Na. I also drop several rows that I do not need, such as the stat and p-value. Here is an example of the result. So I will keep gene ID, base mean, log 2 fold changes, and log fold change standard error, and adjust p-value. Next, I select differentially expressed gene based on the adjust p-value that is less than 0.05 and arrange them in ascending order. For each database, I calculate the number of up and down regulated gene too. It looks like Boolean raised at 25 versus Boolean raised at 19 degrees has the smallest number of differential genes. It fits our prediction since Boolean is an isogenic line, which means every individual flies are genetically identical. So I should see the minimum number of differential genes. To identify genes that are affected by environmental temperature, I look at two datasets, Boolean at 25 versus Boolean at 19 degrees Celsius, and wild-type Cantonese at 25 versus Cantonese at 19 degrees Celsius. For genes that change in both datasets, they are likely to be affected by temperature only. So here, I perform an inner join for differential genes of both datasets. And to identify the common gene name, I add those information from the gene annotation table. And here, I identified 7 genes that are related to temperature shift. Next, I also want to see the genetic difference between Boolean and wild-type Cantonese line. I look at the common genes in these two databases, Boolean versus Cantonese at 25 or at 19 degrees Celsius. There are a total of 355 genes in common. And when I look at the chromosome location, I have some interesting findings. Given that the size of these chromosomes in flies are fairly similar, it is intriguing to see nearly over half of the differential genes are located on the second chromosome, which is interesting that we might want to study further. We can also calculate the number of up or down regulated genes in this Boolean. Here are example of the top 20 genes that most significantly differentially expressed gene. A lot of them have a CG number only, for example like here, CG, suggesting that they are novel genes, and we would like to, and that would be interesting to characterize them further. Finally, I also want to identify the genes that are related to the bully phenotype. Since only bully raised at 25 degrees Celsius has clear hyperaggressive phenotypes, I identified genes that are common in two databases, the bully versus wild-type Cantonese at 25 and bully at 25 versus bully at 19 degrees Celsius. To avoid the effect of temperature shift, I removed genes that also exist in Cantonese 25 versus Cantonese 19 degrees Celsius database. This gives me a total of 7 genes that are related to the hyperaggressive phenotype. In addition to Spark, I also use Neo4j, a graph database, to analyze the data. First, I generate the data file for nodes. It contains all distinct differential genes in 4 datasets. Here's an example. It has gene name, gene ID, and chromosome information. Next, I generate the data file for edges. It contains gene ID and whether the gene is upregulated or not. In Neo4j, I create node experimental nodes and edge. Here's an example for one experimental dataset that is a B25 bully grow at 25 degrees versus bully raised at 19 degrees Celsius. The gene nodes are in purple, experimental node is red here, and there are links between the genes and the experiment whenever it is identified as differentially expressed. To find genes that are affected by temperature, I identify 7 genes using 2 experimental datasets, the C25 Cantonese 25 versus Cantonese raised at 19 degrees Celsius, and bully raised at 25 versus bully raised at 19 degrees Celsius. In this graph, I see that 4 of them are downregulated and 1 of them is upregulated. There are 2 genes that are upregulated in one dataset but downregulated in the others. Since they are not consistent, they might not be good candidates. So I end up with 5 genes in this case. Using the same approach, I try to identify genes that are related to the hyperaggressive phenotype in bully. I found 7 genes in total that are differentially expressed in both the bully raised at 25 and Cantonese raised at 25. And bully raised at 25 versus bully raised at 19 degrees Celsius. But they are not expressed in the Cantonese 25 versus Cantonese 19. 3 of them are downregulated and 2 of them are upregulated. I also found that 2 genes that are inconsistent and probably not relevant to this phenotype. For conclusion, I successfully used Spark and Neo4j to identify differentially expressed genes. I identified 5 genes that are affected by temperature. There are a total of 355 genes that are differentially expressed in bully, and 5 genes that are linked to the hyperaggressive phenotypes.